Welcome to this deep dive. So today our mission is to completely unpack this really groundbreaking paper that came out in May 2026 in the journal Cell.
Yeah, it's a massive one.
It really is. It's titled Unbiased Niche Labeling Maps, Immune-Excluded Niche in Bone Metastasis. And it comes from Zhu Liu and their team at Baylor College of Medicine. We're going to explore the source material, the experimental models, the clinical data, basically everything they use to uncover this wild survival mechanism that tumors use to conquer your skeletal system.
Right, and the central premise of this paper it completely upends our traditional understanding of metastasis. I mean, when we normally think about cancer spreading, the real biological challenge has always been studying the host microenvironment.
Like the soil, right?
Exactly. What we call the soil in the seed and soil hypothesis. Because to actually cure bone metastasis, we first have to see exactly what the cancer is interacting with in real time, which is incredibly hard.
Yeah, because I think, most of us naturally picture cancer as this solitary, hostile invader, like an alien ship landing on a new planet, completely isolated, just shooting at everything in sight.
Yeah, that's the common perception.
But when you actually step into the world of metastatic microenvironments, you realize that alien ship isn't fighting alone at all. It has somehow convinced the local planetary defense force to build it a fortress. And that is exactly what we are investigating today for you. How cancer's secret weapon to survive in the bone is actually our own corrupted immune cells.
It's terrifying but fascinating. And to understand how that fortress is built, the researchers, they had to solve this massive logistical problem first, because the interactions between a metastatic cancer cell and the host body's native cells are just incredibly transient.
Transient, like how fast are we talking?
We are talking about fleeting physical handshakes that last fractions of a second, just a quick bump. And standard proximity labeling tools in biology, like the BioID or APEX systems, they often require hours of continuous contact to successfully label a neighboring cell.
Oh, wow. So if they just bump into each other for a millisecond, the old tools won't even register it.
They're simply too slow. It's like trying to photograph a speeding bullet with a really slow shutter speed on your camera. You just miss these rapid metastatic interactions entirely.
Okay, let's unpack this. Because if the traditional tools are too slow, how do we illuminate this invisible microscopic battlefield where the combatants only touch for a millisecond?
Well, the researchers had to invent a completely new technology. They call it SAMENT, which stands for Sortus A-based microenvironment niche tagging.
SAMENT.
Yeah, and they actually looked to nature for inspiration here, specifically to bacteria. Sortus A is normally this enzyme that bacteria use to anchor proteins to their own cell walls.
Yeah.
And it acts incredibly fast. So the researchers hijacked this bacterial enzyme to create a localized split-second labeling system.
Okay, let's break down the mechanics of this SAMENT flashlight, because this is cool.
Yeah.
How do you engineer a human cancer cell to use a bacterial enzyme?
Right, so it requires a highly engineered sender and receiver framework. The sender in this scenario is the cancer cell. The researchers genetically modified these cancer cells to express a specific variant of the enzyme called MGSRTA.
Okay.
And here is the really crucial design element. They fused this enzyme to a GFP nanobody.
Let's pause on the nanobody for a second, because I know that's an important structural detail for you listening.
Yeah.
We hear a lot about regular antibodies, which are these large Y-shaped proteins. But a nanobody is much smaller, right? Derived originally from the immune systems of alpacas and llamas.
Yeah, exactly. Alpacas and llamas have these incredibly compact antibodies, and that size is essential here. When two cells touch, the synaptic cleft between them, the actual gap, is microscopic. A bulky, traditional antibody just wouldn't fit properly to facilitate a transfer.
It would be like trying to fit a couch through a dog door.
Right. The nanobody fits perfectly. Now, for the receiver side of the equation, the surrounding host niche cells, the native cells already in the mouse's body, they are engineered to express surface GFP.
Which is green fluorescent protein, right?
Exactly, a protein that sits right on their outer membrane.
So if the cancer cell bumps into a host cell, the nanobody on the cancer cell seeks out and locks onto the GFP on the host cell. Does that just anchor them together forever, creating a permanent bridge?
No, not quite. The physical interaction is still completely transient. They touch and eventually separate. The nanobody just holds them close for a fraction of a second. But, and this is the genius part, in that split second, the MGSRTA enzyme goes to work.
Like a molecular stapler.
Exactly like a molecular stapler. It takes a substrate circulating in the system called biotin LPTGS. which is essentially a 5-amino acid chain carrying a biotin molecule. And it covalently attaches or staples that biotin tag onto the surface of the receiver cell.
Okay, so the interaction is temporary, but the tag itself is permanent. It's literally like the cancer cell is walking through a crowded room with an indelible marker, leaving a biotin stain on anyone it physically bumps into.
Yeah.
But wait, why use biotin specifically?
Because biotin has this exceptionally high natural affinity for a protein called streptavidin. By tagging the neighboring cells with biotin, the researchers created a highly reliable molecular handle.
Oh, I see.
Yeah, later on, they can take the tissue sample, pour it over a bed of streptavidin, and only the tagged cells will stick to it. It allows for perfect isolation.
But there has to be a way to control when this marker is actively writing, right? You wouldn't want the cancer cell tagging everything it touches while it's just, traveling through the bloodstream long before it even reaches the bone.
Spot on. The system requires precise temporal control. To achieve this, they installed a genetic switch known as a TET-on system.
Okay.
So the entire SAMENT tagging mechanism only activates when a specific drug called doxycycline, or DOX, is administered to the mouse.
Oh, wow. So they just wait.
Exactly.
Yeah.
This gives the researchers the power to let the cancer spread naturally, wait until it colonizes the bone, and then flip the DOX switch by giving the mouse the drug. That lets them map the exact neighborhood at a highly specific moment in time.
Okay, let's do a quick recap for you listening to anchor this first concept before we look at the actual results.
Good idea.
So bullet 1, metastatic interactions are super fleeting. Standard tools miss them. Bullet 2, the SAMENT system solves this by acting as a time-controlled molecular stapler, using that nanobuddy and a sortase A enzyme to permanently tag any host cell that physically touches the tumor. Bullet 3, it allows scientists to definitively separate the tagged neighbors from the non-tagged cells later.
Perfectly summarized.
Yeah.
So here's a question for you listening to think about. Imagine having this tool. What would you expect to find surrounding a metastatic cancer cell? Because I know my first thought, if we isolate these cells, I'd assume we're going to see a lot of exhausted T cells, immune soldiers that tried to fight the tumor but just got overwhelmed.
Right, that would be the logical assumption. But the sequencing data revealed something much more drastic. Once they injected these engineered cancer cells, specifically they used LLC1 lung cancer cells and B16 melanoma cells into the mice and flipped that doxycycline switch, they isolated the tagged cells.
What did they do with them?
They ran single-cell RNA sequencing on them, and they mapped the neighborhoods in the bone, the brain, the liver, and the lung. And the T cells weren't exhausted. They were virtually absent.
Wait, absent as in the cancer's immediate neighborhood is just totally devoid of the immune system's primary assassins.
Completely. Across all 4 of those distinct organ microenvironments, the tagged neighborhood was uniquely depleted of T cells. The tumor had effectively created this exclusion zone.
That is wild.
Yeah, but the space directly touching the tumor wasn't empty. It was highly enriched with 2 specific cell types: endothelial cells, which form the blood vessels to basically feed the tumor, and most prominently, macrophages.
Okay, here's where it gets totally counterintuitive, because macrophages are white blood cells whose name literally translates to big eaters.
Yeah.
Their primary biological role is to patrol the body, identify foreign debris or invaders, and physically engulf them.
Right. They eat the bad guys.
So if the immediate vicinity of the tumor is packed with macrophages, why aren't they eating the cancer? How are they acting as the bouncers for this immune-free safe room?
To answer that, the single-cell RNA sequencing provided a really crucial clue regarding the maturation of these cells. In the general untagged bone marrow space, you see a healthy population of monocytes.
Those are the circulating precursors to macrophages, right?
Exactly. They are like the Wookiees. But in the tagged population, the cells directly touching the tumor, those monocytes were severely depleted. Meanwhile, mature macrophages were heavily enriched.
Oh, which implies the tumor isn't just like passively sitting next to existing macrophages. It is forcing the precursor monocytes to rapidly differentiate the moment they come into contact.
Yes.
But how does mere physical touch trigger such a rapid maturation?
It comes down to localized metabolic and signaling reprogramming. The physical proximity allows the tumor to alter the immediate concentration of metabolites and signaling molecules. It alters them so drastically that the monocyte essentially has no choice but to mature into a specific, highly suppressive type of macrophage.
It brainwashes them.
Basically, yeah.
Okay.
The tumor is actively reprogramming them to serve as its personal bodyguard detail.
Okay, let's do our second quick recap for you listening. Bullet 1. SAMENT mapped the metastatic neighborhood in multiple organs. Bullet 2. The universal finding was that T cells are locked out entirely. Bullet 3. Macrophages completely dominate the space directly touching the tumor. So the question we have to ask next is, what exactly is the tumor whispering to these macrophages to make them turn rogue? What is the tumor doing to the internal wiring to turn them into such effective bouncers?
To decode that internal wiring, the researchers zoomed in specifically on the bone metastases. They took the single-cell RNA-seq data and they compared the transcriptomes.
The complete gene expression profiles.
Right. They compared the transcriptomes of the biotin-tagged macrophages against the untagged regular macrophages that were just hanging out in the same bone.
So they basically compared the corrupted bodyguards to the regular cellular police force. What was the defining genetic difference?
The tagged macrophages show this massive, highly unusual activation of the ESR1 gene.
Wait, ESR1, that is the gene that codes for estrogen receptor alpha, right?
That's it.
I have to admit, it's genuinely fascinating to encounter this here. Estrogen receptors are like classic targets when we talk about luminal breast cancer cells. But finding them profoundly upregulated inside a host's myeloid immune cell, inside the bone marrow, that feels like a massive biological leap. Why would a macrophage need an estrogen receptor to protect a tumor?
It's a huge leap. It represents a significant paradigm shift in how we view the tumor microenvironment entirely. Because this isn't the cancer cell utilizing estrogen to proliferate. This is the host's own immune system utilizing an estrogen receptor in response to the cancer.
Wow.
And the researchers uncovered the exact mechanical cascade that causes this. It all starts with lipid metabolism.
Let's trace that causality, because we know tumors are essentially metabolic black holes, right? They drastically alter the concentration of nutrients, including fatty acids, in their immediate vicinity. Are the macrophages importing these altered lipids?
They are. The close physical contact with the tumor fundamentally alters the lipid profile inside the macrophage. And macrophages contain a specific nuclear receptor called PPAR-gamma, which functions as a primary lipid sensor.
PPAR-gamma. That is a well-known hallmark of M2 macrophages, right? The wound-healing immunosuppressive variant.
Correct. So when these specific tumor-altered lipids enter the macrophage, they act as activating ligands. They physically bind to PPAR-gamma.
And then what happens?
Well, once activated by those lipids, PPAR-gamma translocates to the nucleus. It moves in and acts as a transcription factor, literally turning on specific genes. And in this highly specific context of tumor contact, the primary gene it upregulates is ESR1.
So the lipid sensor directly forces the macrophage to produce estrogen receptor alpha. Did they provide, like, in vitro proof of this specific link? Because that sounds like a bold claim.
They provided a very high level of mechanistic evidence.
Yeah.
The researchers took macrophages in a laboratory dish, completely isolated from any tumor cells, just in a dish, and treated them with a drug called rosiglitazone.
Okay, what is that?
Rosiglitazone is a known potent synthetic agonist for PPAR-gamma. It artificially stimulates that receptor. And when they applied the drug, it directly and dramatically upregulated the ESR1 gene in those macrophages.
Wow. Okay, let's use a chain reaction mnemonic to lock in the cellular logic for you listening. Touch triggers fat, fat triggers estrogen.
I like that.
Yeah, so the tumor's physical touch alters the macrophage's internal lipid or fat pathways. Those altered lipids activate the PPAR-gamma receptor, and PPAR-gamma flips the switch to flood the macrophage with estrogen receptor alpha. Touch triggers fat, fat triggers estrogen.
That cascade is exactly what drives the pathology. And once that estrogen receptor alpha signaling network is active, it prompts the macrophage to secrete this toxic cocktail of suppressive factors. These factors actively prevent T cells from recruiting to the area or activating if they somehow do manage to arrive. The macrophage just becomes a perfect impenetrable shield.
Okay, let's hit our third recap to solidify this mechanism before we look at the clinical impact. Bullet 1. Tagged macrophages in the bone express high levels of ER-alpha. Bullet 2. The tumor contact alters lipid metabolism via PPAR-gamma. Bullet 3. PPAR-gamma activation turns on the ESR1 gene, equipping the macrophage with ER-alpha.
That's the sequence.
So for you listening, ponder this. If this specific estrogen receptor is the linchpin of the cancer's defense, what happens when we use a genetic scalpel to just pull that pin out? Break the shield entirely?
Well, to test that exact hypothesis and reach the highest level of experimental evidence, the researchers didn't just observe the shield, they engineered a way to actively destroy it using a conditional knockout mouse model.
Right, and let's clarify that. Because if we just bred a mouse with a systemic knockout of the ESR1 gene, like the whole body doesn't have it, it would cause massive biological chaos.
Yeah.
Estrogen receptor alpha is required for bone density regulation, brain function, cardiovascular health. If you knock it out everywhere, the bone microenvironment would be so disturbed you couldn't possibly isolate the macrophage's specific role in metastasis.
Exactly. That is the exact methodological trap they had to avoid. You wouldn't learn anything specific. So to isolate the variable perfectly, they utilized the Krelok's recombination system, specifically driven by the lyasm promoter.
Walk us through how that acts as a targeted scalpel.
Sure. So the lyasm promoter is a sequence of DNA that is really only active in myeloid cells, like macrophages. The researchers engineered mice so that an enzyme called Cre recombinase is only produced when that lyasm promoter is active.
Okay, so Cre is only in the macrophages.
Right. Then they took the ESR1 gene and flanked it with specific genetic tags called LOXP sites. The Cre recombinase enzyme basically acts like a pair of scissors that specifically cut the DNA between those LOXP tags.
Oh, that's brilliant. So because the Cre are only produced in macrophages, the ESR1 gene is perfectly intact in the mouse's brain and heart and regular bone cells. But inside the macrophages, the gene is sliced out. They are physically incapable of building this estrogen shield.
Exactly. And the precision of that model yielded truly dramatic functional results. When they introduced the metastatic cancer cells into these specific knockout mice, the immune-excluded niche completely collapsed.
Wow.
Yeah, without the ER-alpha signaling in the macrophages, CD8-positive T cells flooded back into the metastatic space. They could finally recognize and attack the tumor.
And what was the ultimate effect on the cancer's ability to survive in the bone?
Bone colonization was severely impaired. Stripping the macrophages of their estrogen receptors functionally crippled the tumor's ability to establish a foothold.
So we have proven the mechanism in mice. But, mouse biology, while foundational, isn't always a one-to-one map to human biology. Did the team bridge that gap with human clinical data?
They absolutely did, and honestly, the clinical data reveals the most profound implication of the entire study. They accessed human clinical databases and analyzed single-cell RNA sequencing data from actual patients suffering from bone metastases.
Naturally, one would assume they looked at breast cancer metastases, given the heavy involvement of estrogen receptors.
They did analyze human bone metastases originating from breast cancer, yes, but they didn't stop there. They also analyzed bone metastases originating from lung cancer and from renal cell carcinoma, which is kidney cancer.
Wait, lung and kidney cancer. But those are not traditionally considered hormone-driven cancers in the way luminal breast cancer is.
That's the kicker. The data showed that it doesn't matter. They found the exact same ER-alpha-positive macrophages dominating the immediate tumor microenvironment across all of these distinct human cancers.
That implies this isn't just a quirk of breast cancer pathology. It is a universal survival tactic.
Yes.
Once a malignant tumor, regardless of the organ it originated in, once it decides to colonize the skeletal system, it exploits the exact same macrophage estrogen loophole to build its fortress. The cancer uses the native logic of the bone's microenvironment against the body.
That's beautifully said. To synthesize this entire journey, the researchers built the SAMENT technology to capture transient cellular interactions that were previously invisible to us. And this tool revealed that tumors monopolize their neighborhood by forcing precursor cells to rapidly differentiate into macrophages.
Right.
Zooming in, they discovered that the tumor alters lipid metabolism to activate PPA or gamma, which upregulates estrogen receptor alpha in those specific macrophages. And finally, by selectively knocking out that receptor, they proved that breaking this mechanism strips the tumor of its armor, allowing the immune system to flood in and fight the bone metastasis across completely varying cancer types.
Okay, time for our final recap. Let's distill this clinical evidence down for you listening. Bullet 1. Knocking out the ESR1 gene, specifically in macrophages, shatters the tumor's defensive shield, letting the T cells back in. Bullet 2. Removing this receptor stops the cancer from effectively colonizing the bone. Bullet 3. These exact same ER-alpha-positive macrophages are present in human bone metastases across multiple, very different types of cancer.
A completely universal tactic.
Which fundamentally changes how we view the battlefield. And it leaves us with a final paradigm-shifting question for you to mull over.
It's a big one.
For decades, modern medicine has utilized potent anti-estrogen drugs like tamoxifen or fulvestrant, primarily to starve breast cancer cells of the hormones they need to proliferate. But based on the mechanisms we've unpacked today, could we one day use those exact same endocrine therapies as immunotherapies?
It's totally possible now.
Right. Imagine a future where a drug traditionally designed for female breast cancer is routinely prescribed to treat a man battling metastatic lung cancer in his bones. Not to attack his cancer directly, but simply to strip the estrogen armor off his corrupted macrophages, allowing his own T cells to finish the job.
It changes everything.
It really does. Next time you read a headline about a breakthrough targeted therapy, take a moment to ask yourself, who is the drug really targeting? Is it attacking the disease itself, or is it hunting the traitor hiding in the neighborhood? Thank you for joining us on this intensely detailed exploration of the microscopic world, and keep questioning the science around you.